So this is part 3 of chapter 13 on meiosis, and we left off talking about the three main types of sexual life cycles. So the third type is what most fungi and some protists use, and in this case gametes fuse to form a zygote, which is the only diploid phase. And then that zygote undergoes meiosis to produce a haploid cell, and then those haploid cells divide by mitosis to form either a unicellular daughter cell or a haploid multicellular adult organism. And then the haploid adult will produce gametes again by mitosis. So again, the only diploid stage in these species is that single-celled zygote. So looking over meiosis among multicellular eukaryotes, animals, and humans, we have the different stages here, and we're going to go through each one. So we have what we call interphase 1, and this is actually the only interphase that will occur during the meiosis process. Then we have prophase 1. And in this case, we have our 2n equal to 4 here. And so our cell is diploid at this point. Then we have metaphase 1 and then anaphase 1. And all of this here, this top row, is what we consider meiosis 1. This second portion here is what we call meiosis 2. So after anaphase 1, what we will have is now a telophase and cytokinesis 1, where we had the division of this cell into two daughter cells. So we have this telophase 1, and this is now where we have our movement from diploid to haploid. So our two daughter cells here as we begin meiosis 2 are now haploid cells. They have half the number of chromosomes. So you can see that if you're doing your count here, we have 1, 2, 3, 4 chromosomes here, and down here each of these cells only has 2 chromosomes. So we have prophase 2, metaphase 2, anaphase 2, and then telophase 2, and then our final cytokinesis. So again, it's important to remember we have the same PMAT process going on that we had in mitosis, but the only place that we have interphase is in the first meiotic set of reactions here. So this is our abbreviation. We have this, remember back from biology, the IPMAT, which we used for mitosis. And this time we have IPMAT, and then that's followed by just a simple PMAT. And so this PMAT is actually the one that's closest to mitosis, because in PMAT is when we have the sister chromatids lining up on the metaphase plate here, and then the sister chromatids being pulled apart, just like we have in mitosis. So we end up going from this haploid and ending up with resulting cells that are also haploid, daughter cells that are haploid. So again, we have this double division idea of meiosis. And DNA replication only occurs in the first of these sets of divisions. So the first division, before it even occurs, is when DNA is replicated, and that's during interphase of the cell cycle. You can see the diagram on the side there with the homologous pair of chromosomes in the diploid parent cell. So we only have one set of chromosomes. And then they go through DNA replication so that we have now the sister chromatids. So again, this is only going to occur before the first meiotic division. And the way that's easy to separate the two different types of division, so after we have DNA replication, we have our first division of meiosis. And the purpose of this first division is to separate the homologous pairs. So remember, these two are homologous pairs. And in our first division, the main goal is to separate out one homologous chromosome in one cell and the other homologous chromosome in the other cell. So if our original cell actually had 4 instead of 2 chromosomes, you would see where we have one homologous, these are homologous pairs and these are homologous pairs. And they're being separated here. So one of this set ends up in one cell and one of this set ends up in this cell. Then the second division of meiosis, the purpose is now then to divide up these sister chromatids. So here we have a set of sister chromatids and we want to separate those out. And these are the 4 daughter cells or gametes that we end up with at the end of meiosis. So I'm going to go through a little bit here, these different stages of meiosis. Oh, and we're going to have sound effects. But basically, a lot of it's going to sound familiar. So this first, let's go back here. This first set of divisions or steps of meiosis 1 are the reduction portion of meiosis. So remember, this is where we're going in meiosis 1 is when we're going to go from diploid to haploid. So let's see here. Let's focus in on some of the things we want to be thinking about here. Okay, let me get to the right slide. Okay, so the key thing here with interphase, again, is when we have the chromosomes being duplicated. We have our nuclear envelope still present. We have chromatin present, invisible, but we do have the chromosomes being duplicated. And then we move into meiosis 1 with our first prophase. So in prophase 1, what we're going to have happen is our homologous chromosomes are going to actually pair up. And this pairing up process, we're going to go into a lot more detail. But this pairing up of homologous chromosomes, so again, this is one from the father, here's one from the mother. This set of homologous chromosomes pairs up in prophase 1 to form a tetrad. The other part of prophase 1 is similar to mitosis, where we have a spindle formation and the centrioles moving to different portions of the cell, different ends of the cell. We'll talk about chiasmata in a little bit. Now in metaphase 1, we have these are our homologous chromosomes in their tetrad formation, and they're lined up on this metaphase plate. So we have our homologous chromosomes in their tetrads lined up, and we have one of the homologous pairs facing one pole of the cell, and the other homologous pair is facing the other pole. And that's important how they're lined up, because as we move into anaphase 1, you're going to see that the kinetochords are going to be guiding and pulling and separating these tetrads and separating out our homologous pairs, so that each of our daughter cells is going to end up with one of the homologous chromosomes. And again, this is how we end up with a haploid cell at the end of anaphase. Okay, so now with meiosis 2, we have the beginning of it with the beginning of telophase 1, where each half of the cell has now a haploid set of chromosomes, And each chromosome still consists of two sister chromatids. So you can see that here. These are two sister chromatids. But we only have now a haploid set of chromosomes. So we have the formation of the cleavage furrow, we have the division of the cytoplasm occurring, and then we end up with our two daughter cells. And our two daughter cells immediately go into prophase 2. So in prophase 2, we have a spindle apparatus being formed again, and the chromosomes, each still are composed of the two sister chromatids, are starting to move towards the metaphase plate. It's important to remember here that we don't have any more replication of chromosomes occurring. So these sister chromatids are just continuing on this process. In metaphase 2, we have the sister chromatids, or each chromosome lining up on the metaphase plate, with one chromatid facing one pole and the other chromatid facing the other. So now it's important to note, and we'll talk about this a little bit later, but at this point these chromatids are not identical because something occurred in prophase 1 and back in meiosis 1 called crossing over, in which genetic information was traded between the homologous pairs. So it's important, and we'll talk about why that is a little later on. But as we move into anaphase 2, we have the pulling apart of the sister chromatids, so that separation of sister chromatids moving into two individual cells. So in telophase 2 and cytokinesis, we again have a cleavage furrow forming, and now we have haploid daughter cells forming. And just like in mitosis, we have nuclei form and the chromosomes begin to decondense. The cytokinesis separates the cytoplasm, and each of these now 4 daughter cells is genetically distinct. It's individual and different from each of the others. And they're also different from the parent cell. So let's stop and look at what crossing over involves. So back in prophase 1, during meiosis 1, we have the duplicated homologs pairing up to make the tetrads. And another way, or another term for these tetrads is a synaptomal complex. How's that for a mouthful? So we have, again, a pair of homologs, one from each parent, that are made up of sister chromatids. And these join up together, actually, and the sister chromatids attach along their length with protein cohesins, which hold them together through the process of these different phases of meiosis. And then we also have some attachment going on of this synaptomal complex. And because of this close alignment of these homologous pairs here, these homologs, there's some twisting that occurs, they believe, actually, and this is still a lot of theory, but in this attachment and close alignment, they actually get tangled up, and there's a cutting of DNA that occurs at the same spot on these homologous chromosomes. And so it's actually going to be one chromatid from one of the homologs and one chromatid of the other homolog. And they end up exchanging pieces of DNA. And it happens to be genes or a portion of the DNA that's exactly the same and coding for the same information or characteristics in each of these different chromosomes. So they're somehow, and they're actually still doing a lot of study, and they're